Massachusetts General Hospital MGH investigators have identified differences in gene transcription within key immune cells that may distinguish those individuals infected with the hepatitis C virus HCV who develop chronic infection from those whose immune systems successfully clear the virus. As they describe in a paper published in the journal Immunity, these changes appear soon after initial emergence of the T-cell response and long before conventional T-cell testing or clinical symptoms indicate whether or not an infection will become chronic. Our primary finding that cellular energy metabolism is dysregulated at the transcriptional level very early on in chronic infection harbors important clues for how we might want to think about the T-cell response to HCV infection and about T-cell exhaustion in general, says lead author David Volsky, Ph.D., a postdoctoral fellow in the MGH gastroenterology unit, who performed this work during his graduate studies. It gives us a potential avenue for finding earlier predictors of disease outcome and, more importantly, tells us what may be causing the failure of the T-cell response in chronic infection, giving us new opportunities to try and intervene before failure occurs. HCV differs from other chronic viral infections such as HIV or hepatitis B in that the infection spontaneously resolves in a small but significant minority of infected individuals, around 20%, within 6 months of infection. While the adaptive immune system responds to HCV in individuals who develop chronic infection-producing cd8 and cd4 T cells specifically targeting the virus, the number of virus-specific T cells eventually diminishes, and those that remain lose their functionality, a phenomenon called T-cell exhaustion. Acute HCV infection is usually mild and non-life-threatening, but the persistence of the virus can lead to chronic, potentially fatal liver diseases such as cirrhosis or liver cancer. Until recently, drugs to treat chronic HCV have had limited effectiveness, but the directly acting antiviral drugs that have become available in recent years are able to cure the infection in more than 90% of patients. However, given the high number of infected people worldwide and the fact that successful treatment does not prevent reinfection in high-risk groups, such as those who inject drugs, development of an effective HCV vaccine remains extremely important. Principal investigator Georg Lauer, MD, Ph.D., MGH Gastroenterology Unit, explains that the infection's unique property of naturally leading to two distinct outcomes, chronic infection versus complete viral clearance, inspired their investigation into the immune system factors leading to protection or viral persistence in human patients. This was only possible because of the availability of a group of 43 individuals diagnosed with HCV soon after their initial infection who provided a total of 78 blood samples 1 to 3 from each that were taken within 36 weeks of infection. Such patients are very difficult to identify, but the MGH group has one of the largest groups in the world, developed through the efforts of co-authors Arthur Kim, MD, MGH Division of Infectious Diseases, and Leah Luis Ximenez, MD, at the Oswaldo Cruz Institute in Rio de Janeiro, Brazil. Of those 43 individuals, the infection resolved in 21 and became chronic in 22. Samples from participants in whom the infection persisted were further divided into two subgroups: chronic, in which infection persists even though the CD8 T cells continue to recognize the circulating HCV viral strain, and escape, in which the CD8 response is rendered blind because the virus has mutated in the region recognized by the T cells. Studying patterns of gene expression in these two populations of chronic-associated T cells enabled the investigators to distinguish between expression changes related to T-cell receptor-mediated signaling and McCaw Prize for Excellence in Community Service. In August 2017, the MGH was once again named to the honor roll in the U.S. News World Report list of America's Best Hospitals. Disclaimer: AAAS and Eureka Alert are not responsible for the accuracy of news releases posted to Eureka Alert by contributing institutions or for the use of any information through the Eureka Alert system.